Two plants, same seed bank, the exact same genetics. Yet one grows strong, healthy, and resilient, while the other stays small, weak, and barely survives. So what's the difference? It's not their DNA. It's the environment around them. In today's video, I'll show you how the environment can literally turn your plant's genes on or off without changing a single letter of their genetic code. Welcome back to the Genetics Growers channel. This is where we explore genetics, cultivation, and everything that helps your plants reach their full genetic potential. And if you're looking to learn even more, be sure to visit our website. Our blog is packed with in-depth articles on cultivation, pest management, the latest agricultural trends, practical step-by-step guides, and plenty of other resources designed for growers like you. You'll find the link in the description below. Now, let's dive into epigenetics. Let's start with the basics. Genetics is the set of instructions every plant carries from the moment it emerges from the seed. Its DNA defines its potential, its characteristics, and its natural limits. Think of it as the plant's instruction manual. But an instruction manual can be used in different ways. You can highlight certain pages, skip others, or save some for later. That's exactly what epigenetics does. It doesn't rewrite the manual. It changes which sections are read, when they're read, and how strongly they're expressed. The word epigenetics comes from the Greek prefix epi, meaning above or on top of. Literally, it means above genetics. These are biological mechanisms that sit on top of the DNA and determine which genes are switched on and which remain switched off, without changing the DNA sequence itself. And this isn't just a concept that belongs in a research lab. It's happening in your grow every single day. The light your plants receive, the temperature, water stress, the nutrients in the growing medium, even physical stimulation from wind or contact with neighboring plants. All of these factors can influence which genes are expressed. In other words, the environment is constantly communicating with your plant's DNA. To make this crystal clear, think of genetics as a computer's hardware. The hardware is fixed. It comes from the factory and determines the machine's maximum potential. Epigenetics, on the other hand, is like the software. It's the programs that are turned on or off depending on what's needed at any given moment. The hardware never changes, but the software determines what actually runs. Here's another way to think about it. Imagine a full orchestra with every musician and every instrument ready to play. That entire orchestra represents your plant's genome. Now, imagine the conductor deciding which instruments play the loudest, which stay silent, and exactly when each one comes in. That's epigenetics. The orchestra doesn't change, but the music does. And that explains something every grower has seen at some point. Two plants, the same genetics, the same seed bank, the same expected phenotype, yet completely different results at harvest. It's not that the genetics failed. It's that the environment conducted the orchestra differently for each plant. Now, without getting too technical, let's look at how this actually works inside the plant. There are three main epigenetic mechanisms. The first is called DNA methylation. This process involves tiny molecules known as methyl groups attaching themselves to specific regions of the DNA, almost like little tags. When one of these tags is added, the gene is switched off or its activity is reduced. When the tag is removed, the gene can become active again. The second mechanism is histone modification. DNA doesn't float freely inside the cell. Instead, it's wrapped around proteins called histones, much like thread wound around a spool. When the DNA is wrapped tightly, the genes are hidden and can't be read. When it loosens, those genes become accessible and can be activated. The third mechanism involves small, non-coding RNAs. These tiny RNA fragments act like precision messengers, selectively silencing specific genes in response to environmental signals, such as a pest attack or a sudden drought. The key thing to remember is this: None of these three mechanisms changes the DNA sequence itself. No genes are erased. No genes are rewritten. They simply determine, layer by layer, which parts of the genetic instruction manual are read out loud and which ones remain silent. Now, let's get to what matters most for us as growers: the environmental factors that are turning genes on and off in our plants every single day. Let's start with light. The amount, quality, and duration of light your plants receive activate genes involved in photosynthesis, flowering, and even the production of pigments and aromatic compounds. A change in the photoperiod doesn't just shift the plant into a new growth stage. It actually reprograms which genes are active at that moment. Next comes water, or more accurately, the lack of it. When a plant experiences water stress, it doesn't simply suffer. It activates an entire network of defense genes. Its stomata close, its roots become stronger, and it begins producing protective compounds. It's a programmed response, triggered epigenetically by water scarcity. Temperature has a similar effect. A sudden cold snap, extreme heat, or sharp swings between day and night temperatures can activate genes responsible for thermal tolerance. In some cases, these events even leave behind a kind of memory that prepares the plant for similar stress in the future. The soil and the nutrients available in it are also powerful epigenetic switches. The availability of nitrogen, phosphorus, potassium, and micronutrients activates entire metabolic pathways, determining whether the plant prioritizes vegetative growth, root development, or the production of flowers and fruit. And here's something that surprises many growers: the soil microbiome. The invisible community of bacteria and fungi surrounding the roots also communicates directly with the plant's epigenetic machinery. Certain beneficial microorganisms can activate defense genes before a real threat even appears, essentially preparing the plant in advance. Even mechanical stress, such as wind, plants rubbing against one another, or physical handling during pruning, triggers a response known as thigmomorphogenesis. The plant adjusts its growth, becomes more compact, and strengthens its tissues, all by activating specific genes. And that brings us to one of the most fascinating discoveries of recent years. Plants have memory. Not memory like ours, of course, but a kind of epigenetic record of what they've experienced. A plant that has gone through moderate drought can become epigenetically marked, allowing it to respond faster and more efficiently if it encounters drought again. This is known as priming, or stress priming. It's one of the reasons why some growers deliberately expose seedlings to carefully controlled levels of mild stress. Not to harm them, but to train them epigenetically before they face a more serious challenge. But here's what's even more remarkable. In some cases, this epigenetic memory can be passed on to the next generation. In other words, seeds produced by a plant that experienced certain environmental conditions may develop with a different epigenetic predisposition, even before they encounter those conditions themselves. This doesn't replace classical genetics or the importance of selecting high-quality seeds. Instead, it adds an entirely new layer to our understanding. The environment experienced by the parent plant can influence how its offspring perform, even though the DNA they inherit remains exactly the same. So, what do you do with all of this information as a grower? First, understand that environmental management isn't just about basic plant care. It's an active tool for influencing gene expression. Every decision you make about lighting, irrigation, nutrition, and temperature is literally helping determine which genes are switched on in your plants. Second, consider using controlled stress priming. Small, carefully managed periods of mild stress, never severe stress, can prepare your plants to become more resilient when they face future challenges, such as sudden weather changes or pest pressure. Third, pay close attention to environmental consistency during critical growth stages. Unnecessary abrupt changes can trigger epigenetic responses that work against you, leading to slower growth or uneven flowering. The goal isn't random stress, it's intentional management. And finally, if you produce seeds, Remember that the conditions in which your mother plants are grown may have epigenetic effects on the next generation. An optimized growing environment doesn't just benefit the current crop, it may also benefit the plants that come from the seeds it produces. To sum everything up, genetics defines a plant's maximum potential, but epigenetics determines how much of that potential is actually expressed and when. Mechanisms such as DNA methylation, histone modification, and small non-coding RNAs act like molecular switches that the environment is constantly turning on and off. Right now, as your plants grow, light, water, temperature, nutrients, the soil microbiome, and even physical contact are actively reshaping their patterns of gene expression. And in some cases, that epigenetic information may even be passed on to the next generation. Understanding epigenetics isn't just a fascinating scientific concept. It's a practical tool that can help you become a better grower. If you'd like to dive deeper into this topic, as well as pest management, step-by-step growing guides, and the latest trends in agriculture, be sure to check out the blog on our website. You'll find the link in the description below. If you found this video helpful, give it a thumbs up, subscribe to the channel, and turn on notifications so you never miss our upcoming videos. And let us know in the comments, which environmental factor do you think is having the biggest impact on your plants right now? We'd love to hear your thoughts. Thanks for watching, and we'll see you in the next Genetics Growers video.